Hi, my name is Morgan Sayanta, and I am going to be explaining gene editing with CRISPR babies. So, a little background because a lot of people haven't actually heard of this. This is Dr. Key Janku, and he genetically altered embryos using CRISPR. He genetically modified two twin babies to be immune to HIV, and the twins Lulu and Nala were born in October of 2018. So I did my news story on the Wall Street Journal, and it said that he spent 2 years making all of this possible, and he was the first person that genetically edited babies. Gene editing trials that are involving like terminally ill adults are still okay to do, and they are still ongoing. But editing embryos is a very controversial topic, and this is because it gets passed down through generations, and you don't really know what to expect through it. They scouted and they actually found dads who are HIV positive who wanted to have babies with moms who are HIV negative. And so he would take this father's blood test that was HIV positive and fake it to make it seem like it was HIV free. And then he would alter the genes before and like without the doctor knowing, and they would be implanted into the mothers. He hired an embryologist to edit the genes at a completely different hospital, and the embryologist was completely aware of the situation, understood that it was illegal, and he kept it a secret from the hospital he was working at and to the doctor that was actually implanting the embryos into the mothers. Getting into CRISPR a little bit, the gene editing tool CRISPR-Cas9 does raise a lot of ethical questions, which I've stated earlier. And it's not really that accurate. It can alter other genes and impair other functions, which was found true in the twins. They found out that they were genetically smarter and than the natural human, and that they were more prone to getting infectious diseases, which was very weird to me. An international guideline, 2017, said that they did not completely ban implanting edited embryos, but it was only for medical reasons if there was no other alternatives. And in this case, with these HIV babies, there is other possible alternatives, which is why Dr. He was in the wrong during this experiment. Dr. He began using CRISPR-Cas9 on monkeys, mice, and non-viable human embryos in 2016. He eventually switched over to doing it on humans. So another question I was wondering was like kind of why he did it. And his initial research was he wanted to find an editing gene that would protect people against familial hypocholesterolemia. But then he went to a village that showed a ton of people who were infected with HIV and were being discriminated against in schools and jobs. And he decided that he wanted to help them instead. After publishing his findings, he was asked by a group of people what were his methods, how did he recruit patients, and did he tell them of the risks? And Dr. Key's response was that he didn't know how to answer these questions. So for my primary article, I have Dr. Key used CRISPR to change both copies of the CCR5 protein. With this change, he wanted to hope that HIV would not infect the white blood cell, but he had a total of 5 women and 13 embryos implanted into the females to hope that some of them would catch. 2 of these women got pregnant, and one of them actually gave birth to a set of twins, which was the twins I just talked about in the beginning. The other woman's child and her health are unknown. We haven't heard anything about what happened to the third kid. The twins were born in the end of October and were deemed healthy by Dr. He in November. So the primary article, I just wanted to show a little example of the CCR5 co-receptor. So as embryos, they were injected with CRISPR construct, and it causes a 32 pair deletion on chromosome 3, resulting in another protein that's actually naturally found in some humans. But instead of this, he made changes to the CCR5 gene. Like it would lead to the production of the non-functional protein. So these changes have never ever been seen in humans before, and some functions of the CCR5 protein are unknown, but we do know that they're located on T cells and white blood cells. He explains that one of the twins was actually heterozygous too. So, and one of the twins, he only took out one of the pairs, and which means that their T cells still express CCR5 proteins, but just in reduced amounts. Accuracy and explanation. So the news article was somewhat accurate because it does explain some of the steps that did occur, but it's not 100% accurate just because we haven't found He's actual research. Only he and his team truly know what happened, and information regarding his own research is completely unavailable to the public. And that's because it's extremely illegal and they don't want anyone doing it again. We can't ask any more questions too because Dr. He has not been seen or heard from since November of 2018, and there's been no updates. He was supposed to be arrested, but now he is just missing. Are some of my citations, and I thank you so much for listening, and I hope you learned something from my presentation. Thank you.